Good afternoon, and thanks for everyone for being here on late afternoon on day 2. Appreciate it. Who we are a biotechnology company that's focused on developing advanced therapeutics for patients with diseases of the intervertebral disc. So we have a very therapy-specific focused technology versus a platform, and I'll take you through that and really what this means. Our company is in its late preclinical phase, and I'll talk more about our timeline in a bit. So the problem here is painful discs in our patients here. So we'll talk about primarily degenerative disc disease, but in a normal, healthy spine, you can look at the image at the top. You have, essentially 4 main components. You have the nucleus pulposus, which breaks down the annulus fibrosus, which is like the casing, the outer plies of a tire. You have the end plate, which is cartilage, where nutrition comes into the disc from the vertebral body because the disc is not fed by a blood supply like other tissues in the body, much like articular cartilage or the cornea, for example, and the vertebral bone. With degenerative disc disease, you have a breakdown of a number of the components within the disc, primarily the nucleus pulposus. Then that impacts the annulus and also the end plates. So you have a progressive disease that does not heal itself in patient subjects. Back pain is what we're attempting to treat. On a global level, it's extremely significant. The incidence rate here is anywhere from 12 to 30% of the population, and pretty much any time I speak to anybody, they know somebody either in their family, friends, or people they work with that are disabled by chronic back pain related to degenerative disc disease. And it has a huge implication on the healthcare system. It's estimated that in the U.S., approximately $100 billion annual spend in the U.S. to treat patients related to this, and that includes some loss of work and productivity. On a personal level, for patients who suffer from this, really it manifests itself in a chronic pain and disability where they have a hard time doing the normal activities they enjoy doing, and it can even impact work, lost wages. And increasing problem, we've heard more about this in the past year, driven by celebrities and various things, is addiction to opiate pain medications. That is one of the methods of treating the chronic pain, so we hope to alleviate that. So from an anatomical perspective, what is degenerative disc disease? So there's some images on the right-hand side. The first column I'll refer you to is really histology of the spine that shows you from the top a healthy segment of a disc. So you can see a very robust white solid center. The annulus looks very good. Everything's in good health, and provides the proper spacing of your spine. So your spine is basically a load-bearing column with mobility built into it. And I'll use the analogy of a shock absorber in your car or truck. When it's new or operates properly, you're able to support the car, the stability, the handling of the vehicle, and absorb shocks. When the disc starts to break down, the disc will start to collapse. The disc height will reduce. On MRI, the disc that comprises of the extracellular matrix with a hydration fluid called proteoglycan starts to dry up and will start to collapse. So if you look at images B down through D, you'll see a progressive degradation of the extracellular matrix in the dish. That cushion, which keeps the disc height maintained and maintains stability, breaks down, and you'll see the disc becoming what we call a dark disc. So the hydration goes away. So you'll get cracks and little fissures and defects in the annulus, and then you'll get things called innervation, where little nerves will go into the disc that will become additional pain generators. The chemistry of the disc changes. It becomes very acidic, so you have cytokines that will trigger pain within the disc. So it becomes this process where it just proliferates, and we can't stop that ourselves within our body. It's very progressive and cyclic, and what I mean by that is that once you have it, you're diagnosed with this chronic and degenerative disc disease, it does not self-correct. So a number of things can cause it. It can be through hereditary genetic predisposition, an injury. You can have an injury that damages it. There can be chemistry changes in the body that can also impact that. But in mild stages, when you're first diagnosed, the first thing normally is over-the-counter nonsteroidal anti-inflammatory drugs, physical therapy, yoga, chiropractic care, and then when those things don't seem to alleviate the symptoms, opiates will come in, and then epidural steroid injections, usually done in a course of 3, to attempt to mitigate some of the inflammation and pain that's going on. And then when the patients get into a moderate phase, there's really not any additional treatments that can be done at that point. So they're really buying time, continued progression. And then at the end stages, if severe disability, they're having neuropathy or radiculopathy, pain in the legs or numbness, then a fusion procedure, which can be very costly. And the challenge with fusion procedures is that one of the manifestations there is adjacent level disease, where you can have wear on the disc or degeneration above and below. So you have this cascade that can occur. So heart of our technology, and we're a company that's in the late preclinical phases, is our Discogenic cells. So that's the engine essentially within our technology, and it is a combination device, our first product, which I'll talk about shortly, because it utilizes the cells in the scaffold. But it's therapeutic progenitor cells that are derived from adult intervertebral disc tissue. So we have a unique approach. It's a differentiated stem cell progenitor, so it isn't an MSC that's sorted. And it comes from a tissue that the disc is comprised of. So we believe that has an advantage of that lineage to what these cells do, and I'll talk about that shortly. So the cells are produced in a GMP environment. They're cryopreserved and scaled in a single donor lot to treat many patients. We currently have 24 issued patents and 11 more in progress right now that cover the methods and applications. So what are these cells? They have a unique characterization. As I mentioned, they're technically a progenitor because they are differentiated stem cell. They have unique properties, and one of the key ones is the extracellular matrix production that these cells have. So coming out of the culture process in our final phase of culture, they produce the two key extracellular matrix components that the disc is actually made of, and that's aggrecan and collagen 2. So these things go in, and they're right away putting out this matrix inside the disc. So the cells don't have to go in there and signal to discover what they need to be and differentiate. So this allows us, we believe, to use fewer cells, and with the potency, they don't have to be in there very long. The second thing, because they do have stem-like properties as a progenitor, they do have the anti-inflammatory effect that is common with an MSC. So those are the key two things relative to MOA we've seen so far. They are non-tumor forming, extremely safe. I'll talk about that shortly. And it is allogeneic, so we can take it from one donor process and go into a number of human subjects. In all of our animal studies, and I'll touch on preclinical, we utilized human discogenic cells in all of our animal models without having to pre-treat the animals, without any type of pathologic issues whatsoever. So very good safety profile of this. They also are very robust. They survive cryopreservation very well from both pre-freeze through post-thaw. So they're very robust, hardy cells. We're able to produce them in a reproducible and GMP-compliant manner. We currently are able to produce these in a phase 1 cGMP manner, and we've been working for a while on our phase 3 scale-up processes, looking ahead to our further regulatory pathway and commercialization. We already have in place within our phase 1 process, key process controls and final release testing, things that include safety, identity, purity, and potency, and reproducible assays that we have right now. We've shown excellent comparability from our R&D runs through a whole series of cGMP runs we've done that takes into account donor variability and various factors with reproducible, consistent results with that. And we've been able to consistently show we're getting the same surface markers for cell identity throughout the process so far. So the first therapeutic candidate that we'll be filing for our IND in the U.S. Basically early second quarter next year, and in Japan also with the PMDA, is what we call our Injectable Discogenic Cell Therapy, or IDCT. So this is a treatment that's interventional. It's an injection for patients that suffer moderate to mild degenerative disc disease. It's cost-effective, non-surgical. The nice thing about this procedure that it's delivered in the same manner as a physiatrist or pain interventionalist that was doing an intradiscal injection or doing a discogram procedure. It uses the same type of spine needle. It's all done in an outpatient setting, an exam room with a spine table with a C-arm. The practitioner places the needle under fluoro. The product itself, combination device, includes a cryofrozen vial of the cells that are thawed at point of use. there's a transfer syringe that contains a mild hyaluronic scaffold, and then it's mixed, and then it's connected to the syringe and injected. So it's a very quick procedure done under local anesthesia. The two images down there basically show how it works. So the cells go in, as I mentioned, they have anti-inflammatory properties, and the matrix deposition, and I'll share preclinical data, but what we hope to see, if we're consistent with what we've done in our preclinical work, is that the new matrix deposition will cause a regenerative effect, increase disc height, the same we've seen in the preclinical work. We're going to have follow-on indications. The first IND and clearance will approve the manufacturing process, and the first device IDCT for a lumbar indication. Follow-on indications will be for herniated disc, we call it SDCT, and then we'll have additional IDC indications for adjacent level treatment and then for cervical. We've seen very good results in our preclinical data. We've worked through 3 species of animals, and over 13 independent studies since 2012, and we've looked at a number of things, time points, both subacute, acute, and chronic. On efficacy, we've looked at a number of key measures on activity. One of the key things we've seen consistently is disc height restoration. So I touched on this restorative effect earlier. So in all the animal models, we work to establish a defect, and that defect is essentially implemented with a needle that's put into the healthy disc of the injured control animals, and the needle causes a puncture, and there's some aspiration, and that causes within 2 weeks a breakdown of the extracellular matrix, and it reduces the disc height on these animals. So it simulates what happens with the human condition. Then at 2 weeks, we'll dose the animals, and we've run a series of dose analysis studies in these animals, and what we've seen consistently across all 3 species in there is that within 4 weeks of injection, we're able to get back between 80 to 90% of the original disc height of these animals based upon regenerative activity of the discogenic cells. And I'll show you some images of that shortly. On safety aspects, we've looked at histology of the spine, clinical pathology, anatomic measures. We just completed our final GLP pivotal study, anatomic pathology, and everything came back phenomenal. And all the clinical observations have been good and consistent through all the animals we've tested. So regenerative effect. Here I'm going to show some histology. So I'm not sure how familiar everyone in the room is here. I know some of you are. We have 3 columns of images here, and we've stained this in 3 different ways in an attempt to show normalized tissue versus abnormal tissue. The first column is an uninjured control. So these were control animals that weren't injured. So it shows you, the upper image down to the bottom one, what are healthy sections of that animal disc. So you can see they have good tissue all the way through, in all 3 images. The center is an injured control. You can see where the disc height is reduced on these. You can see where instead of the solid bright image in the center of the disc, you can see, it's darkened. And then on the right are section of animals that were treated with IDCT, which essentially looks the same as the uninjured control. So we've been able to show histologically, pathologically normalized acellular matrix tissue that's formed in these animals that really is attributed to the improvement we see. And the cells are transient. We've tracked the cells, and we find that they're gone within 3 weeks in the subject animals. So this is a pivotal year for us, no pun intended. We're transitioning to clinical trials. We're right now completing the final phases of our pivotal studies. We've had multiple meetings with the US FDA and the Japanese PMDA, so we'll be submitting our IND and then CTN between end of first quarter, early second quarter. So we're looking forward to getting to that point. And we're completing the final studies and then the workup of that data early in first quarter. And then, pending everything going well, we're looking at starting our trials in the second half of 2017 in both of those 2 markets. We've prioritized our global market strategy based on a number of things. One is market size, the potential for it. Our patents, we've made it a point to make sure we've secured IP coverage in the key markets that match that. Also, the regulatory environment, as everyone here is aware and looking at, is key. So looking at that both for clinical processes, how you can expeditiously work with the regulatory bodies to get into the trial process. Manufacturing requirements, our goal is to try to be consistent where we produce CGMP process at different phases that will satisfy the different global bodies. And reimbursement. We're trying to interrupt the disease continuum, and here this is based upon a study that was done in spine. The typical treatment pathway is for the patient to get 2 years of conservative care, followed by 3 epidural injections, and then what we found in this study was that half the patients within 2 years of completing that went on to a spinal fusion procedure. So we would intervene early after the patient goes through 2 months of conservative care or 3 months. We would then intervene with a single injection of IDCT, and this would take a lot of costs out of the system and treat the patients earlier when they can have a best response and save cost. The market's robust. We've looked at this. We started looking at codes back in 2013, and we've looked at IC9 codes, and then based upon the different sections of the market for herniated disc, degenerative disc, and then the epidural steroid injection market, it's a nearly 15.2 million market potential in the U.S., and 2x that internationally. So large market based upon the incidence rate. We're a private company. We're currently fundraising. We've raised more than just a little over $10 million. That has gotten us up to the point of our final preclinical phase. We're closing tomorrow the first part of our Series B, and we have the second part open that'll fund us well into our interim analysis. In summary, the advantage of DiscGenics and IDCT is we have a very differentiated, focused technology on treating specifically the disc with our Discogenic cells. And once again, that's utilizing source cells that are native to the environment of the disc. So they're programmed specifically to have a regenerative effect and deal with the pain and disability. Scalable process where we have a frozen off-the-shelf process that's delivered in a well-established procedure that people are familiar with, the practitioners are there, and we have robust initial manufacturing, reproducibility, and strong preclinical data with disc height improvement, and a very experienced team and strong expertise. So once again, thanks for listening, and if there's any questions, I'll be around afterwards. Thank you.